the anatomy of the stomach, so we're on page 24.
Do you listen to yourself?
Alright, so this view of the stomach, the lesser omentum, has basically been removed, and you can see the lesser curvature with the left and right gastric arteries. And here's the greater omentum hanging down from the greater curvature. So when we look at the stomach, we're going to just kind of look at the overall structures of it and then look at the GPAT aspect of it. So where the esophagus is attached to the stomach, there is a small thickening of the smooth muscle, the circular layer, which creates our second sphincter. So remember, the first one was the mouth, all right? And this second sphincter is identified as the cardiac sphincter, or if you would like to write more letters, you can call it the gastroesophageal. And this opens up as food is moved down through the esophagus to allow it to enter into the stomach. Okay, it's really that passageway of peristalsis that functions in the relaxation of that sphincter, and food enters into the stomach. So in reference to that cardiac sphincter, we also have a cardia region. So basically that area that I've outlined with the dotted line would be the region known as the cardia. This upper portion here uses a term that we first used with the uterus. So that's the fundus. It's higher than the entrance of food through the sphincter. So if someone is having a taking radioactive milkshake for the purpose of having x-rays done to follow the peristaltic activities of the esophagus or to look for other anomalies. This area often shows up black, while the rest of the stomach is a bright white from the radioactivity flow rate. That's because air is usually present in the fundus. Okay. Most of the rest of this area is the body. And this narrowed region is known as the pylorus. This is the lesser curvature, as we talked about with the lesser omentum. And this would be the greater curvature, where we have the origin of the greater omentum. Okay? So those are the regions of the stomach. If we look at the opening of the stomach, the lining is thrown into these longitudinal folds, identified by a term that we have also used previously with the vagina and the urinary bladder. And these longitudinal folds are known as rugae. So you can see those here in the model of the stomach. All right. Now if we look at the mucosa, the GPAT of the stomach, this is described on page 26. And we can see it in this pattern here. So we now also see a mucosa, a submucosa, a muscularis externa, and a serosa. So let's look at the mucosa first. And if we look at the mucosal plan, kind of a schematic of the stomach and the small intestine and the large intestine, we can see a comparison. So here would be the Here would be a comparison of the mucosal scheme of the stomach, where we have wide gastric pits and narrow gastric glands, kind of an even surface. So that would be the stomach. In the small intestine, we have outgrowths called villi. These would be covered with simple columnar epithelium, and we have intestinal glands. There's a little bit more connective tissue between those. All right, this is where the muscularis mucosa would be. This is small intestine. And then on Thursday, we'll describe the colon, or the large intestine, and that is just glands. So we would have the surface with just even glands without any villi, and this would be the colon or large intestine. Okay, that's kind of the surface pattern that we would see. So with the GPAT of the stomach, let's focus on the mucosa first. So what we see is our gastric pit, branched or unbranched gastric glands, and we've now changed our epithelium, starting with the stomach, to simple columnar. So we see simple columnar epithelia. They obviously will have microvilli, but we really don't see goblet cells. And I want to show you, this is also off of the review slides, but we have a transition between, an abrupt transition between the stomach and the esophagus. So here's the non-cornified stratified squamous of the esophagus, and the boom, we get the gastric pits and simple columnar epithelium of the stomach. So if we look at a higher power view of that, these would be the gastric pits and the gastric glands, and then our muscularis mucosa is right here. Okay. I still have not figured out why I have the trouble with this. So we have several different types of cells that are present. The gastric pit and the upper portion of the gland are mucus cells. Again, these are not goblet cells. They're simple columnar cells, but they secrete mucus. And obviously the stomach, with its low pH, is in greater need of protection because it's a simple columnar layer, not a stratified squamous. And then as we move down into the gastric pits, we see predominantly two types of cells. Cells that secrete digestive enzymes and cells that secrete hydrochloric acid. So these rounder cells that I'm drawing are the ones that secrete hydrochloric acid. And the other cells are identified as chief cells. Right? I'll make those blue. So the chief cells are going to be secreting pepsinogen, like fibrinogen, that's the inactive form of a protein enzyme called pepsin. So we have to The hydrochloric acid functions to activate that by removing some amino acids. And we have the active pepsin. We also have gastric lipase. And remember intrinsic factor? Or the absorption of vitamin B12? So those are predominantly most of the cells. And then we have the parietal cells. Which secrete hydrochloric acid. And this hydrochloric acid works on the pepsinogen on the way out the gastric glands and activates it to pepsin. And change, certainly the pH of the stomach is that really acidic 1 to 2. And then at the very bottom of the gastric glands, we have endocrine cells. So the stomach is a mixed, it's an endocrine and an exocrine gland. So these are known as enteroendocrine cells. Sometimes known as G cells because of the hormone is gastrin. It starts with a G. So these secrete the hormone gastrin. Which is interesting as a hormone, it works on itself. Okay, it's a hormone that changes the activity of the stomach. And then finally at the deep surface, here, we would have our muscularis mucosa. All right, so we have simple columnar epithelium, a loose irregular connective tissue. That's standard. And a muscularis mucosa. Now, depending on where we are, this is kind of incorrect because it shows the chief cells not starting to the bottom. They actually would be starting way up here. So in that particular diagram, it's a little bit off. So this is probably more accurate. So the mucus cells to here and then scattered throughout here. The chief cells and these little orange ones are the parietal cells and the yellow ones are the G cells or enteroendocrine cells. Okay. So this would be the mucosa of the stomach. Now, if you were to become really, really, really tiny and walk through the stomach, its surface would, actually, wrong slide. This is what the surface of the stomach would look like. So you can see the strands of mucus, and these would be the mucus cells, surface mucus cells. These would be the openings of the gastric pits. So that's what's coated by all of that mucus.
The gastric pits are the mucosa?
The gastric pits are those white space, that white space at the top of the gastric gland where it says mucus cells. So this would be the gastric pit here. Okay. So here are two images of the stomach. Here we have really long gastric pits and short gastric glands. Here we have really long gastric glands with the relatively short gastric pits. So if this was a histology class, you could tell if you were in the fundus or in the pylorus or in the body of the stomach based on the proportion of the length of the pit to the length of the gland. All right? You guys just have to worry about whether you're in the stomach or the small intestine. All right, so that's the mucosa. If we then return to the slide here, so the next area we have is submucosa, and nothing different about that. It fits our standard dense irregular connective tissue. Yes.
So is mucosa the stuff in the pit or?
The mucosa, mucus, we have mucus and mucosa, okay? So the mucosa is the layer that goes from muscularis mucosa all the way to the lumen. The mucus cells are part of the mucosal epithelium and they're secreting mucus, okay? So can I erase the diagram for mucosa? All right? The next layer is the submucosa. And that's what every other submucosa has been, with dense irregular connective tissue, larger blood vessels, larger arteries, and so on, nerves. That's what's binding our mucosa to our underlying layer. We have our muscularis externa. And our typical pattern was an inner circular and an outer longitudinal layer. In the stomach, we add a third layer. So here's our inner circular, here's our outer layer, and then we have an innermost oblique. Because the stomach functions in both mechanical, primarily mechanical digestion, and just a little bit of chemical. So that oblique layer provides a twisting motion for the activity of the stomach. All right, so here would be the oblique fibers, and then we have circular fibers and longitudinal fibers. So if this, I'll draw a smaller stomach here that I can put the muscles on. So we're going to have oblique. All right, so that's the innermost oblique, or the deepest layer. And then we have our inner circular. And then running the length of the stomach, we would have our, and that would be covering everything, okay, it's not just on the surfaces. We would have our outer longitudinal. So if we look at this next slide, kind of puts those all together. So here's the innermost oblique. Here's the inner circular. And then these fibers here would be the outer longitudinal. And that allows a twisting, ringing motion that provides the mechanical digestion, the breaking down, the emulsification of the lipids in making it smaller droplets of oil, mechanically breaking down the food for increased surface area. And then finally, we have our outermost layer, which is a true serosa. Again, just our visceral peritoneum. So if we look at this model, this grayish membrane here is the serosa. Okay? This side of it shows some of the circular and oblique and longitudinal layers of the muscle. All right, so the stomach is primarily mechanical digestion. There might be some absorption of lipid-soluble material, primarily alcohol. which is why you drink, it's better to have some food in your stomach. That delays the absorption. It doesn't have quite as rapid an effect, prolonged over time. Plus alcohol is toxic to the lining of the cells. Yes?
That's what I was going to ask. So if you did happen to drink on an empty stomach, is it going to cause enhanced damage to the epithelium itself? Figured as much. I just had to ask in case
Yeah.
I'm missing something.
In fact, when I was teaching at the medical school, one of the professors showed a series of images of the stomach epithelium that had been exposed to alcohol, and it just basically kills off those cells. It's good. Of course, it depends on the concentration of alcohol as well, but
You said it was basically chemical?
yes. So there's very little chemical digestion, which is what we're going to look at now. Okay? So again, turn back this time to page 44. That's the mirror of defining an animal physiology. So funny hopping here. My daughter had a tea rally yesterday, and they started with the oldest kids and then worked to the youngest, which seemed kind of backwards. The youngest were terrified by the time they actually got out there in the day and it was hot. I thought they did it the other way. They could have watched the older kids ride when they were done. And there was, so at the very end, they're just walking over the poles, you know, instead of jumping. But the next to the last, this girl just walked her pony up to the jump, and what is it, propulsion, is that the right word? Up to the jump, and the pony just went, and the poor girl just came right out of her saddle and her stirrups, and then came down sideways and landed on the ground on her hip, and it was like the very, very first jump. But that's what happens at that level. All right, so when we look at the stomach, all right, here's the 3 foodstuffs that are going to be possible to be broken down. And remember the enzymes that I listed from the chief cells that are being synthesized by the stomach epithelium? What were the 2 enzymes? Pepsinogen, activated to pepsin, and gastric lipase. All right, anything for carbohydrates?
No.
No. So carbohydrates are started in the mouth. Will they continue in the stomach? What's the pH of the mouth?
6.8.
6.8, yeah, slightly acidic, sometimes a little higher. The salivary amylase is going to be denatured in the acidic pH of the stomach. So that's going to stop if it ever got started very much, if you chewed long enough to get it started in the mouth. So we don't have carbohydrate digestion taking place in the stomach. We don't have a gastric amylase. And the amylase that is secreted in the mouth becomes denatured and inactivated. So that's not going to take place there at all. All right? We will see a little bit of protein digestion with the pepsin. So again, this is an intermediate. So we have polypeptides. Our protein, which is just long strands of amino acids. The intermediate enzyme available is pepsin. And our products are small peptides and dipeptides. So we call them oligopeptides if it's a few amino acids, and dipeptides if it's 2, obviously. Are any of these single amino acids? No. So no absorption of amino acids in the stomach. We didn't break anything down in the mouth, so we don't have that to occur, and we don't have anything small enough to cross the plasma membrane across the gastric wall. All right? We do have gastric lipase, like we did salivary, but again, we don't have the bile salts. We don't have anything to maintain an increased surface area. So I'm going to put this in parentheses because, again, theoretically, it can occur, but not to any measurable amount. look at the sequences of the control of digestion, all right, I'm going to save that for, I covered it this morning, but I think it'll be less jumpy if I save that for Thursday and we look at both the cephalic, gastric, and intestinal phases, okay, as far as smelling food and having saliva produced and all of that. Kind of tie all that together on Thursday. So when food is in the stomach and it's mechanically churned and moved around, it forms depending on how much juices are in there, a paste or a liquid type of material known as chyme. All right, so that's what moves from the stomach to the small intestine, is that chyme. So that lower portion on page 44 that talks about the stimulation of gastric activity, we'll cover that on Thursday, okay, as we move the food through into the small intestine. So let's jump back to the small intestine on page 32. So the small intestine on page 28. And we've talked about this a lot already because this is the standard by which everything else is compared to. So there are 3 regions to the small intestine. Okay, the last 2 regions functions kind of overlap. But this is where we actually do our serious chemical digestion and all of our nutrient absorption, except for some vitamins and water in the large intestine. The small intestine is about 20 feet, 20 to 21 feet long. And it looks like a coil of reproductively active snakes all moving around in there. So I'm going to give you some hints about where I would tag it because our models aren't colored like this chart is. So the stomach ends at the pyloric sphincter. All right, and that opens up to allow just a tablespoon or 2 of chyme to move into the small intestine at a time, and that provides time for the small intestine, the duodenum, to neutralize and change the pH and mix the contents of the liver and the pancreas. And so that first portion of the small intestine is C-shaped and is known as the duodenum. Between 10 and 12 inches long. It has been fused to the posterior abdominal wall, so it is our first structure so far that can be considered to be retroperitoneal. So it only has serosa on its anterior surface, all right, facing towards the peritoneal cavity. It receives the chyme from the stomach. It also receives content from the liver and content from the pancreas. So that's really its major function. So it's going to receive the bile salts from the liver. So we'll come back and talk about we're going to end with the liver. This is the common bile duct. If there's a lot of fat in the food, bile is going to be released into the duodenum. And then from the pancreas, it's going to receive the rest of the digestive intermediate enzymes and bicarbonate ions. So intermediate digestive enzymes. So we're talking pancreatic amylase, trypsin, chymotrypsin, and pancreatic lipase. All right, so these are made by exocrine cells of the pancreas, and also bicarbonate ions, because the duodenum works at a pH of 8 to 9. So these three components, bile salts, chyme, and pancreatic secretions, all enter into the duodenum. It's short, so there's really no digestion that takes place here of any significance. It's just the mixing, the neutralizing of these contents. So again, we stop the activity of the gastric enzymes because they require a pH of 1 to 2 to work. So now we start using the pancreatic enzymes. Okay. At the end of the duodenum, and I wrote this stuff right where I needed to put something else, we have the superior mesenteric artery and vein crossing from behind the stomach and over the small intestine. So their presence helps to indicate the change from duodenum to jejunum. The other change is the fact that the jejunum is intraperitoneal. So it makes a right-hand bend that's over here. As it comes out into the surrounded by visceral peritoneum. And so this is the beginning of the jejunum. You can see it in this model. So here is the stomach. All right, and the duodenum beginning here curving around. And so that relationship, here is a superior mesenteric artery and vein crossing over the duodenum, and then it makes this right angle turn. Comes out towards the abdominal cavity. So this would be the first part of the jejunum. Okay. On the chart, you can see it right here. Now when it's all coiled up in here, you can't tell, and I can't tell, which part of that model loop is supposed to be jejunum and which is supposed to be ileum, all right? So where I will take the ileum, this is where I would take the jejunum, is up here where you can see its attachment to the duodenum. Where I would take the ileum is where you can see it ending by attaching or joining to the large intestine. So over here we would have colon, and where you can see the ileum attaching to that would be that tag. So both the ileum and the duodenum, the jejunum, let me write that name in, are intraperitoneal. Here's the same diagram I used to show you the blood supply. So here again is the superior mesenteric artery crossing over the duodenum. Here's that bend that begins the jejunum. And then we see the branches of the superior mesenteric artery, and here's the ileum ending as it's attached to the colon. Now histologically, if you were to see a slide, you'd studied and were aware of it, you can tell by the difference in the shape and regularity of the villi, whether it's jejunum or ileum. But you would have to use a microscope for that. Right? you were a surgeon looking at the abdominal cavity or looking at a cadaver, there's a change in the blood supply. So we do most of our absorption from the ileum. These have multiple arcades with short, straight branches going to the ileum, whereas the jejunum has a single arcade and longer branches as a result. So from the vascular aspect of it, that's a unique change that occurs. All right, so let's look at, let's see what do I want here. Let's look at the GPAT now for the small intestine. And remember, this is what the model is based on. This is what the plan is based on, is the small intestine, because it forms the greatest link of our digestive tract. Because of the importance of the small intestine for digestion, that leads to the importance of surface area. So we need a lot of surface area for secretion of enzymes and absorption of product. And the mucosa has our villi. This is lined with simple columnar epithelia. And then we have intestinal glands that extend down to the muscularis mucosa. So this would be a villus, and this is an intestinal gland. The eponym for intestinal gland was crypts of lubricant. Much more interesting to ask on a quiz question than intestinal gland. But as we move away from individuals' names, we move away from the crypts of lubricants. So the epithelium is again simple columnar. Microvilli are still important. But now we add goblet cells instead of having mucus cells. So simple columnar. And I'm not going to do the entire, it's taking forever. So even the ones that don't have lines are still simple columnar cells, okay? And we have goblet cells scattered among them. So that's the source of the mucus. We don't tend to have them in the intestinal glands, but the intestinal glands here, rather than forming the site of secretions for the intestine like the gastric glands did, these function more as an escalator. So the cells that are found in the intestinal glands will eventually be the cells that are on top of the villi. All right, so they kind of just migrate up the surface until they get exposed to the digestive enzymes and get destroyed and replaced by additional cells. So this structure is a villus, in contrast to the cells, which are each going to have microvilli. And we'll talk about what's in there. The unique structure that we add to the mucosa is, this should probably be green, a blind lymphatic capillary known as a lacteal. Those are present only in the small intestine. Okay. So here are the intestinal glands, here are the villi, and then we have our loose irregular connective tissue and a muscularis mucosa.
Is that within this space?
So the loose irregular connective tissue, this is gonna have additional glands, all right? But yes, all of this in here would be loose irregular connective tissue. This yellow material that you see here. So this is a pretty regular picture. It's usually not as regular as this, but here are the villi lined with simple columnar epithelium. The white structures that you see are the goblet cells, and here would be intestinal glands with the muscularis mucosa running along here. Okay. This next layer is the submucosa. So the submucosa is standard dense irregular connective tissue. But two locations we have specialties. Duodenum, we once again see submucosal glands secreting mucus. What do you think the reason for that is? What was the reason we had submucosal glands in the, again, the acidic nature of the acidic nature of the gastric chyme that's coming in. So here is our muscularis mucosa. This is our submucosa, and these are all submucosal glands, all right, secreting mucus in addition to the goblet cells that are present, and we find these only in the duodenum. So when you see villi, all right, you might go, well, those look like gastric pits and gastric glands. We don't have submucosal glands in the stomach, all right, so this is the duodenum. And we'll work on histology on Thursday. I'll give you some more clues about differentiating in those aspects. Another difference in the submucosa that you're already aware of are the increased number of lymphatic nodules in the ileum. So those are found in the submucosa. Remember the Peyer's patches we talked about? So in the ileum, we have the Peyer's patches and lymphatic follicles. So that brings us to the muscularis externa, and we're still on our standard plan. So we make no modifications. We have an intercircular layer. And an outer longitudinal layer. Okay. And then finally, we have a serosa, which is the visceral peritoneum. So this model is of the small intestine, all right? So here's our mucosa with villi, covered with simple columnar epithelium and goblet cells, the blue ovals, intestinal glands. The gray material is the loose irregular connective tissue, and this green structure right here is the lacteal. or lymphatic capillary, which is present in the small intestine to pick up our digested lipids, all right? We'll talk about why on Thursday. Here's the muscularis mucosa and our submucosa, indicating the lymphatic nodules or follicles that are, we find them throughout the digestive tract, but we have multiple ones in the ileum. And then over here on the side are the submucosal glands, representing the duodenum. Okay? So these glands, if you see, it's easier to see them on this side portion of the model. Then we have our inner circular, outer longitudinal layer of the muscularis externa, and finally the serosa, the visceral peritoneum on the outer surface. All right, so I'm going to move ahead to the liver, because we need to know about the liver to do the physiology on Thursday, and then we'll do the large intestine anatomy on Thursday as we follow the food into the large intestine. So jump one more time, but not quite as far, to page 36. And then we'll be done with lecture on Thursday. Oh, I know, there's one more thing I forgot. Sorry. Because of, so this is, back again. These are the 3 orders of folding that are listed on page 29. And then we can move to the liver. This is because, again, of the importance in surface area. So from microscopic to gross, large enough to not need a microscope, we have, the more things are folded, the more surface area you have. So the first order of folding are folds of plasma membrane with the content of actin. So if we look at the surface of one of those simple columnar cells, you can see how much we increase the surface area with the addition of microvilli. Right? Now the digestive enzymes, the ones that do the final chemical breakdown to allow small enough molecules to be formed that will actually cross into the plasma, into the cell, those are never actually secreted out of the cell, but are found within the plasma membrane of those microvilli. So a sucrose, a disaccharide molecule, would bump into sucrase. That would increase the rate at which those 2 sugars are broken apart, and then the glucose or the fructose could be internalized by the cell. All right, so the more microvilli we have, the more of those final stage enzymes are present. So our first order of folding creates the microvillus, which is our singular form. And this is a fold of plasma membrane. Then we take all these columnar cells that are forming the epithelium, and